And we left off talking about this cycle that exists where you're in the cell, where you're constantly breaking ATP down, which releases energy. We said that's the hydrolysis of ATP releases energy, and that energy can be used to power things that require energy in the cell. And then at the same time that's constantly hydrolyzing or breaking down that ATP, the cell needs to be constantly making ATP. And in order to make ATP, you're going to constantly have to input energy into that process. Okay, so that's where we left off. Now we're going to talk about oxidation reduction reactions. So at the very top of the slide, it says another way of transferring energy is to transfer electrons. So the first way of transferring energy that we talked about was through ATP. So you acquire energy in one form, and then you transform that energy into the form of ATP, and then ATP can, when it's broken down, release that energy, and that energy can be used for another process. So that's one way of transferring energy. Another is through transferring electrons. And you're going to get the transfer of electrons through what are called oxidation reduction reactions. And an oxidation reduction reaction is a chemical reaction in which there's a complete or partial transfer of electrons from one reactant to the other. Okay, so in an oxidation reduction reaction, there's a complete or partial transfer of electrons from one reactant to the other. Okay, when you break it down, oxidation refers to the loss of electrons. Oxidation refers to the loss of electrons, and reduction refers to the gain of electrons. So if you're the reactant that loses an electron, then you've been oxidized. If you're the reactant that gains an electron, then you've been reduced. Okay. So an easy way to remember that is just to remember the acronym oil rig. And the letters stand for oxidation is loss, so that's referring to electrons. So oxidation is loss, reduction is gain. Okay, and again, it's either the loss or gain of electrons. Okay, so when you look at this reaction down here, where A is losing an electron and B is gaining an electron, which one is being oxidized in this reaction? Right, A. So A is the one that's losing an electron, and remember oxidation is loss, and then B is the one being reduced, and that's the reactant that's gaining an electron. So what we're going to see is during both cellular respiration and photosynthesis that you have these things called electron transport chains in which electrons are being passed or transferred from one component to the next, so you have a series of oxidation reduction reactions going on. And what happens is as the electrons are passing through the electron transport chain, they'll start off with a high amount of energy, and then they'll slowly lose energy as they are transferred. And then we'll see in this specific case that the energy released during those electron transfers is used to create an electrochemical gradient. Okay. When we study cellular respiration, we'll focus on two molecules that we can think of as electron carriers. So we're going to see that they pick up electrons in the beginning part of cellular respiration, and then at the end part of cellular respiration, they drop the electrons off at the electron transport chain. And those two molecules are NADH and FADH2. So you can just think of those as electron carriers, and they're going to be involved in cellular respiration. This slide, you don't need to memorize the structure of this, but this is showing you, so on the left-hand side, this is NAD+. And then if you reduce NAD plus to NADH, and when you reduce it, it's picking up two electrons. That's the reduction part. It also picks up a hydrogen ion. Okay, so this would be NAD plus being reduced to NADH, and then the reverse reaction is NADH being oxidized back to NAD plus. And we're going to see that during cellular respiration, both happen. So at first, NAD plus picks up electrons when it's reduced, and then when it drops the electrons off, it's oxidized. Okay, so both NADH and FADH2 are going to do that during cellular respiration. Okay, do you guys have any questions about oxidation reduction reactions? Okay, so that covers the energy part of this topic. The next thing that we're going to cover are enzymes. And what we're going to do is watch two animations describing how enzymes function. One just talking about a general enzyme, and then the second video will describe a specific enzyme, and then we'll talk about them more on the other side of the videos.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Okay, so that's giving you a general idea of what enzymes do, and this is to give you a specific example of an enzyme found in your body.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
Okay, so that described enzymes for you. And just in terms of the definition that we're going to use in this class for enzymes, it was at the beginning of the first animation. So an enzyme is going to be any protein that speeds up chemical reactions. Another way to define them is that an enzyme is a protein that acts as a catalyst. Okay, so an enzyme is a protein that either speeds up a chemical reaction or acts as a catalyst. It's important to know how enzymes are going to speed up chemical reactions. And they speed up chemical reactions by lowering something called the activation energy. And so in terms of what the activation energy is, I'll show it on the graph in a second. But the activation energy is the amount of energy that the reactants have to absorb in order for the reaction to start. Okay, so the activation energy is the amount of energy that the reactants have to absorb before a reaction will start. And you can think of the activation energy as an energy barrier that sits between the reactants and formation of products. So you can think of the activation energy as a barrier. So when you're looking at this graph here, this is something that graphs we've seen before. So on the y-axis you have free energy, and then on the x-axis you have the course of the reaction. So you're going from reactants to products. And so what they're showing you here is we're going to look at the red line first. Here's the free energy of the reactants. And then you have this thing, we're not going to get into what it is, but it's called the transition state. And the energy change from the reactants up into the peak here, that's the activation energy. So this is the amount of energy that has to be absorbed in order for the reaction to start. And then once you overcome that energy barrier, then you'll form the products. Okay, and what this graph is supposed to represent, the red line is supposed to represent the reaction without an enzyme, whereas the blue line represents the reaction with the enzyme. So you can see what happens in this case, that you go from this large of an activation energy to this smaller activation energy, and that's as a result of the enzyme. So the enzyme lowers this barrier that sits between the reactants and the products, and that's the way that it speeds up. By lowering that energy barrier, it makes it easier for the reaction to occur, and it happens at a faster rate. Okay, another important thing that they told you in the animation that is important to realize is that enzymes also are not changed by the reaction. And what that means is that after the enzyme catalyzes the reaction, the enzyme doesn't change in any way after the reaction, which means that it can catalyze the same reaction over and over again. So it's never used up or changed by the reaction that's taking place. So that goes back to something that you guys answered last week in last week's the lab you did 2 weeks ago. They asked you the question, do you need to make a molecule of amylase for every molecule of amylose that you want to break down? And the answer to that question was a no. And the reason you don't need to do that is because one molecule of amylase can break down multiple molecules of starch. So what happens is the enzyme will bind the reactant, form the product, release the product, and then once it's released the product, it can bind another reactant and perform the same reaction, and it can do that over and over again. Okay, so that's also an important thing to realize about enzymes. Okay, in terms of some vocabulary that's important to know with respect to enzymes, one they introduced to you in the animation, which was the active site. Another thing that's useful to know is what's called the substrate. So we'll start with the substrate. So the substrate is the reactant or reactants, so sometimes there's multiple substrates. The substrate is the reactant or reactants that the enzyme acts on. Okay, so it's the reactant or reactants that the enzyme acts on. So in the example where there was the enzyme sucrase, sucrase you saw broke down sucrose. So sucrase acts on sucrose, so sucrose is the substrate, whereas sucrase is the enzyme. Okay, so and what they're showing you here in this picture, you don't. This is called hexokinase, but you don't need to remember that. This particular enzyme has two substrates. One of them is ATP, and the other is glucose. And so in that case, that enzyme has two different substrates. And then the active site is simply the site on the enzyme where the substrates bind, and also where the chemical reaction is going to take place. Okay, so again, the active site is a site or a region on the enzyme where the substrate or substrates bind, and then the chemical reaction takes place. And it's important to realize that the active site shows specificity for the substrate. So similar to the fact that we talked about channels and carriers show specificity for what they transport, enzymes show a high degree of specificity for the chemical reactions that they catalyze. And the reason they do that is because their active site is very specific for the substrates that bind to that active site. So as an example, they gave you sucrase in the animation. Sucrose can bind to sucrase, and sucrase can break it down. But then if you take another disaccharide, maltose, maltose cannot bind to sucrase's active site, and so sucrase cannot break down maltose. You have a separate enzyme called maltase that would break maltose down. Okay, so kind of an analogy that's often used for enzymes and their substrates is a lock and key mechanism. So in this case, the key would be the substrate, and then the space that you put the key into would be the active site of the enzyme. And that's the interaction between a lock and a key is very specific, and the interaction between a substrate and an active site is specific in the same way for enzymes. Okay, so in addition to those things, Some enzymes, not all enzymes, will require additional non-protein components. The important thing to realize is that if a enzyme has one of these non-protein components that we're going to talk about, which is either a cofactor, a coenzyme, or a prosthetic group, the enzyme can't act without it. Okay, so if it requires one of those things, then it can't act without it. So a cofactor is simply any non-protein molecule or ion that's required for the proper functioning of an enzyme. So a cofactor is any non-protein molecule, or can be an ion required for the functioning of an enzyme. In terms of ions, specific examples of cofactors would be iron ions and zinc ions are two examples of ions that act as cofactors for enzymes. And then a coenzyme is pretty easy to remember now that we've defined cofactor. So a coenzyme is simply a cofactor that's an organic molecule. So if the cofactor is an organic molecule, so a carbon-containing molecule, then you simply call the cofactor a coenzyme. And then the last one is a prosthetic group. A prosthetic group is different from a cofactor and a coenzyme because it's covalently attached to the enzyme. Okay, so either the ion or the organic molecule is actually covalently attached to the protein, and then it's called a prosthetic group. Okay, just as a side note, information that might be useful for you don't need to know it for a quiz or exam, but the reason that one of the reasons that vitamins are important in your diet is because that many of them act as they either directly act as coenzymes or your body uses the vitamin to make an essential coenzyme. So a lot of vitamins are either used as coenzymes or used to make coenzymes, and that's one of the reasons that they're important in your diet. And this is just giving you several examples of vitamins that are coenzymes in your cells. Okay. Do you guys have any questions so far about enzymes? Okay, so there are some common types of enzymes in terms of their names, and there are certain ones that you have to know for this class. So it's kind of useful to know all of these if you're going to go on to take more bio classes in biochemistry, because if you know these common names, then they kind of already indicate to you what the enzyme might do. So I'll just point out, first I'll point out the ones that you have to know for this class. So there's certain ones that I want you to be able to define. And the definitions are on the slide there for you, but I'll go through them. And the only reason that I want you to specifically know these is because they come up in the class again. So you need to know polymerases. You need to know kinases. You need to know phosphatases, oxidoreductases, and ATPases. So basically from polymerases down. Polymerases, kinases, phosphatases, oxidoreductases, and then ATPases.
Okay, so it's only 5, right?
Yes. Okay. Yeah. So from polymerases down. Okay, so just to connect this with what I'm not going to ask you right now in terms of this topic to list specific examples of these enzymes, but I'll just give you some so you can connect it to things we're going to learn later on in the course. In terms of so a polymerase is an enzyme that it says catalyze a polymerization reaction. What that means is a polymerase is the enzyme that connects together the monomers to form the polymer. So a polymerase connects together many monomers to form a polymer. And we'll study two polymerases in this class. One is called DNA polymerase, which is used in DNA replication. And DNA polymerase takes nucleotides and connects them together to form a strand of DNA during DNA replication. And then the other polymerase is RNA polymerase. And this is going to be used during transcription. And remember, in transcription, you're using DNA as a template to synthesize an RNA molecule. So RNA polymerase is going to be the enzyme that connects together RNA nucleotides to actually synthesize the RNA molecule. So those are just two specific examples of polymerases that we're going to study in this class. Okay, the next one are called kinases. And kinases are a class of enzymes that catalyze the attachment of phosphate groups to a molecule. Okay, so they add a phosphate group to a molecule. That's what a kinase is. And then what happens in this case is the addition of that phosphate group to that molecule affects that molecule's function. Okay, phosphatases do the exact opposite of kinases, and phosphatases catalyze the removal of phosphate groups. So phosphatases catalyze the removal of a phosphate group. And these two types of enzymes are going to come up twice in this class. So in the first case, they'll come up in this topic that we're talking about, because one way of regulating another enzyme's activity is by adding or removing a phosphate group from it. So what you can have in one example is you can have an enzyme that is present in the cell, but it can't work. You can consider it's off. It can't catalyze the reaction. And what will happen is a kinase will come along, add a phosphate group to that enzyme, and then that activates the enzyme, and now the enzyme can catalyze the reaction. And then after the enzyme has added the phosphate group and the enzyme is active, if you want the enzyme to be turned off again, then you have a separate phosphatase that can come to the enzyme, remove the phosphate group, and then once the phosphate group has been removed, the enzyme is inactive again. It's still present, it's just inactive in the cell. So kinases and phosphatases are going to be one way in which another enzyme's activity is regulated within the cell. Kinases and phosphatases will also be involved in the next topic that we're going to talk about when we discuss cell signaling, because we're going to see that one way that a signal inside of a cell can be relayed is through what's called a kinase cascade. And what happens in a kinase cascade is, let's say you have molecules A, B, and C. Molecule A adds a phosphate group to B, then B adds a phosphate group to C, and so on. And so the signal is being relayed by the addition of phosphate groups. And then you can see that you could turn that signal off through a phosphatase by removing the phosphate groups. But the one thing that both of those have in common is that when you add or remove the phosphate group from the target molecule, it affects the function of that molecule. So then you also have, and we're not going to talk about any specific kinases or phosphatases, but you should know what they do in terms of those two things that I just talked about. Another class of enzymes that you should know are oxidoreductases. And these also have other names. They can be simply called just reductases or dehydrogenases. And these enzymes catalyze oxidation reduction reactions. So oxidoreductases catalyze oxidation reduction reactions. And we'll see two examples for these. We'll see one called the pyruvate dehydrogenase complex. This will be involved in cellular respiration. But the pyruvate dehydrogenase complex, what it does is it catalyzes the oxidation of pyruvate. And then we'll also see another enzyme that's involved in photosynthesis that's called NADP+ reductase. And this enzyme reduces NADP+. Okay, and then the last enzyme that you should know in terms of common names is ATPases. And these are enzymes that catalyze the hydrolysis of ATP. So an ATPase is an enzyme that catalyzes the hydrolysis of ATP. And one of the things that this says on the slide there is that there's actually a wide range of proteins that contain an ATPase as part of the protein. and this would be the energy harvesting part of that protein. So you've already actually seen an example of that in membrane function, structure, and transport. We talked about the sodium, potassium, ATPase. But when we talked about it, we discussed the fact that it was an antiporter, so it's a carrier that transports both sodium and potassium against their concentration gradients. But we said that that carrier gets the energy to do that by hydrolyzing ATP, and this part of the carrier has an ATPase on it, and that's the part of the carrier that actually hydrolyzes ATP and allows the carrier to harness the energy from that hydrolysis. And then, so there's a lot of proteins in your cells that have ATPase activity to them, and they're used to harvest the energy from ATP hydrolysis. Okay, do you guys have any questions about the common enzymes that you need to know for the class? Okay, so the next thing is more of a, I don't know if it's conceptual, but so this is showing you the pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex is actually a what you call a multi-enzyme complex. So what that means is that this is showing you one pyruvate dehydrogenase complex, but that one complex consists of multiple enzymes. Okay, so the one complex has actually multiple enzymes that are part of it. You don't need to remember the specific number for this one, but there's actually 3 different enzymes that are part of this complex. Each enzyme catalyzes a different reaction, but all those enzymes are bound together as a complex. You also have multiple molecules of the same enzyme within one complex. Okay, for this part of the course, it's important to realize that multi-enzyme complexes are common in cells. And one thing that multi-enzyme complexes do is they speed up the rate at which a process is going to happen. So enzymes themselves speed up a chemical reaction, but a multi-enzyme complex is going to speed up the rate at which a whole process takes place. So the enzymes that make up a multi-enzyme complex are not just arbitrarily bound together. They're all involved in a common process. So, for example, pyruvate dehydrogenase complex is involved in oxidizing pyruvate, and that's in order to do that, it actually takes 3 separate reactions. Each reaction is catalyzed by a different enzyme within the complex, and the reason that this speeds it up is rather than having enzyme 1 catalyze the first reaction, so enzyme 1 acts and produces a product that enzyme 2 then needs to bind. So rather than having enzyme 1 producing the product needing to find enzyme 2 making product number 2, and then product number 2 having to find enzyme 3, what can happen in a multi-enzyme complex is the first enzyme just passes the product directly to the second enzyme can pass it directly to the third, and by being bound together in that way, that's going to speed up the rate at which that reaction happens, or those 3 separate reactions happen. So multi-enzyme complexes will speed up the rate at which a process occurs. Okay, do you guys have any questions about multi-enzyme complexes? Okay, so what you're looking at in this, you can think of this in 2 ways. It's a basic reaction path for an enzyme catalyzed reaction, or you can think of it as the steps in an enzyme catalyzed reaction. Okay, so either a basic reaction path for an enzyme catalyzed reaction or the steps of a basic enzyme catalyzed reaction. So what you have in this picture is E represents the enzyme, S represents the substrate, and then P is going to represent the product. So what you have here is in the first step, you have the enzyme and the substrate have to bind together. So the first step is the enzyme binds the substrate. And that's going to form a complex that's specifically referred to as the enzyme substrate complex. So that's on the slide there. But the enzyme with the substrate bound to it is called the enzyme substrate complex. So then the next step is that the reaction is catalyzed by the enzyme, so substrate gets converted into product. And then the last step is that the product needs to be released from the enzyme. And then once the product's released, this enzyme can go back and bind another substrate molecule and do the whole thing over again. But essentially you have binding of the substrate, and then conversion of the substrate to product, and then release of the product. Okay. So in terms of studying enzymes, we're going to talk about two measurements that you can make for an enzyme that will give you some clue or give you some information about the enzyme. And the two parameters that you can measure for every enzyme, one is called Vmax, and the V refers to velocity. And then the other one is Km, and Km is specifically going to be a particular substrate concentration. So when we say velocity for an enzyme catalyzed reaction, what we're talking about is how quickly is substrate converted into product. Okay, so when you talk about velocity, you're not talking about how fast something is actually physically moving. You're talking about how fast does substrate get converted into product. How quickly does that happen with the enzyme? And Vmax is specifically the maximal velocity at saturating substrate concentrations. Okay, and what that means is that the way that you would do this assay to determine Vmax is you would have several separate test tubes, each with a constant amount of enzyme in it, and then you would add increasing concentrations of substrate. At some point, your enzyme is going to become saturated with substrate, and what that means is that all the active sites in that test tube for all the enzyme molecules are occupied by substrate. So when it's saturated, all the active sites are occupied by substrate. And so the Vmax is the velocity of the reaction when that's the case. So when you've reached saturating substrate concentrations. And what that simply tells you is how quickly can the enzyme catalyze the reaction. Because previous to Vmax, another thing that's going to add into the time in which it takes to catalyze the reaction is, well, how quickly does the enzyme and the substrate find each other and bind? But at saturating substrate concentrations, you don't have to worry about them finding each other. It's saturated, and so you're simply measuring how quickly the enzyme can catalyze the reaction. The other parameter that you should know is Km. And Km is simply the concentration, it's the substrate concentration at which you reach 1/2 your Vmax. Okay, so Km is the concentration of substrate at which you're going to reach 1/2 your Km. Sorry, yeah, 1/2 the Vmax. So the substrate concentration at which you reach 1/2 your Vmax. Sorry about that. Okay, so what that, and in terms of what can that indicate for the enzyme? So one thing, if you have a low Km, what that means is that you reach your maximal velocity at a very low substrate concentration. Okay, so if you have a low Km, that means you reach, you're going to reach your maximal velocity at a low substrate concentration. And a low Km generally, not always, but generally indicates that the enzyme binds the substrate very tightly. Okay, so enzymes can have varying what's called affinity for a substrate. And if the Km is low, that generally indicates a very high affinity for the substrate, meaning that they bind together very tightly, versus a low affinity, meaning that they don't bind together very tightly. So a low Km generally indicates a very tight binding.
What do you think is responsible for that, since they're practically specialized to fit with each other?
Well, it specifically has to do with the active site and the substrate and how the interaction between the two. Okay. So those are just two parameters that you can measure to determine, or sorry, two things that you can measure in a lab about an enzyme, and then it indicates to you something about the enzyme. Okay, so then the next thing that we're going to talk about are two types of inhibitors of enzymes. So one is going to be called a competitive inhibitor, and the other is going to be called a non-competitive inhibitor. And so a competitive inhibitor is an enzyme inhibitor that inhibits an enzyme by binding to the active site. Okay, so a competitive inhibitor is an inhibitor that inhibits the enzyme by binding to the active site. So the reason that it's called competitive is because it competes with the substrate. Okay, so a competitive inhibitor competes with the substrate, and they're competing for the active site. Yep.
So it inhibits the substrate?
Well, the way it inhibits the enzyme, but what it inhibits then is it prevents the substrate from binding to the active site. So if the substrate occupies the active site, then if the inhibitor occupies the active site, that means the substrate can't bind there. And if the substrate doesn't bind the active site, then the enzyme can't catalyze the reaction.
What's the point of inhibitors then?
What?
Why do they inhibit that?
There's in terms of inhibitors, they occur naturally as a way of regulating enzyme activity. So again, in some cases you're going to want the enzyme on. In other cases you're going to want the enzyme off. And one way of keeping an enzyme off without destroying it would be through a competitive inhibitor. In terms of outside of natural, well, it would still be natural, but they could be poisons or also drugs. So certain drugs and certain poisons can act as competitive inhibitors of enzymes. An example of that sarin gas is I don't know if it's a competitive inhibitor, never mind. But it inhibits an enzyme, but I can't remember if it's competitive or not. But anyways, a competitive inhibitor will inhibit, I'll give you examples later on in the course once we learn about specific enzymes and specific pathways. So I can only give you specific examples once we study those. But a competitive inhibitor inhibits an enzyme by binding to the active site. A non-competitive inhibitor also inhibits an enzyme, but it does that by binding to a site on the enzyme that's not the active site. So a non-competitive inhibitor inhibits an enzyme, and it does it by binding to a site that's not the active site. You can call that other site, in this case that the inhibitor binds to, an allosteric site. And we'll go on to talk about what allosteric regulation is in a few minutes. But the site where the non-competitive inhibitor binds is called the allosteric site. Okay, and one way that this can happen is that when the inhibitor binds to the enzyme, it causes a change in conformation of the enzyme, and that change in conformation or change in shape could cause the enzyme to no longer recognize the substrate because it changes the shape of the active site. So that's one way that a non-competitive inhibitor, by binding to one side of the enzyme, that could translate to inhibiting the active site of the enzyme.
So since they don't have to compete, like it could stop one that's already catalyzing a reaction.
Mm-hmm. One thing that you don't need to, one of the questions that well, I'll wait till we get to it in the lecture questions, and then I'll point it out. Okay, so those are two types of inhibitors. The next thing that we're going to talk about are two environmental factors that you already know from lab affect enzyme function, and that's going to be temperature and pH. And every enzyme has an optimum temperature range, and every enzyme also has an optimal pH range. And what I told you in lab was that the optimum temperature range and optimal pH range for an enzyme is going to be the temperature and pH range that it's normally found at. Okay, so as an example of that, the optimum temperature for for enzymes in your body is about 35 degrees Celsius to 40 degrees Celsius, and body temperature is 37. Okay, so all your enzymes better be able to work within this range because your body is going to, the temperature is going to fall in that range, and that happens to be the optimum temperature for most of the enzymes in your body. In terms of most organisms, the optimal pH is between 6 and an 8. So the optimal pH for most enzymes is going to be between 6 and 8. That's because most biological fluids fall within that range. You already know of 2 exceptions. One from lab. They asked you what the optimum pH for an enzyme secreted into the stomach was. And an example of that is pepsin. And it has an acidic, it has a optimum pH that's acidic. And that's because that enzyme needs to function in an acidic environment. The other exception that you know, we talked about lysozymes that contain hydrolytic enzymes that break down various macromolecules. And we said that on the id side of the lysosome, the pH is about a 4.8, and all the enzymes in a lysosome have an optimum pH of around 4.8, and because that's the natural environment they're found in. Okay. Do you guys have any questions about, and what's one thing that can be happening to an enzyme either as the temperature goes up or the pH changes that can affect its ability to catalyze the reaction? Right, denaturation. So remember, as you heat is something that can cause a protein to denature. So as you increase the temperature, you could be denaturing the enzyme, and then it's no longer going to function. And then the same thing when you change the pH, that can also lead to, in either direction, that can lead to the protein denaturing and then not working anymore. Okay. So the next thing we're going to study is how cells actually, so a cell is not going to regulate enzyme activity by changing its pH or changing its temperature, because that wouldn't make sense. You're not going to change the whole temperature inside the cell or change the whole pH of the cell just to regulate one enzyme. Okay, so what we are going to talk about next is how do cells regulate the activity of individual enzymes. There's actually several common ways that enzyme activity is regulated. And what we're talking about when we say regulate enzyme activity is, can the enzyme catalyze the reaction, or is it being inhibited from catalyzing the reaction? Okay? And one way of regulating enzyme activity is called allosteric regulation, which we'll go into first. I already mentioned to you the addition and removal of phosphate groups, and this is the one where kinases and phosphatases are involved. Then we'll talk about proteolytic activation. And then the last two we won't really talk about in much detail, but I'll just mention them now. So one way of regulating enzymes is by keeping them in compartments. So for example, you keep all the hydrolytic enzymes in a lysosome. And if you keep an enzyme in a particular compartment, what you can do is you can regulate the enzyme's access to substrate, or you could regulate the release of product from that compartment. Okay, so in terms of keeping it in a compartment, you can regulate 2 things. It's access to substrate. So if you want something broken down, you have to deliver it to the lysosome. And then the release of product can also be controlled from a compartment. And then obviously enzymes are proteins. So a cell, if it wants an enzyme to be made, has to synthesize that protein. And that's one way of regulating it, is by upregulating the enzyme's synthesis. By upregulate, what I mean is you start producing the actual enzyme that you want. And then you can also specifically mark an enzyme for destruction. So you mark it, and then the cell destroys the enzyme. And obviously, if the enzyme is destroyed, then it can't function anymore. Okay, so those last 2 we won't really go into much detail, but the other 3 we'll go into a little bit more detail. So the first one is allosteric regulation. And allosteric regulation in terms of enzymes. So allosteric regulation is something that can actually happen to other proteins that are not enzymes. So the definition that I'm going to give you for allosteric regulation is the definition for enzymes. So an allosteric regulation is when you have a molecule that binds to a protein at one site, and then that binding affects the function of the active site. Okay, so you have the binding of a molecule to one site on the enzyme, that one site being not the active site, but the effect of that binding is that it affects the function of the active site. Okay, and allosteric regulation can be both positive and negative. So in some instances of allosteric regulation, when the small molecule binds to the site on the enzyme, that's going to end up activating the enzyme or turning it on. In other cases of allosteric regulation, you have a molecule binding to the enzyme, and that ends up turning the enzyme off. Okay. And just as an example of allosteric regulation that's on the side here, so this is an enzyme that has two shapes. It has a shape in which it's on and able to catalyze the reaction, and then a shape when it's off. And then this nucleotide CTP, when it binds to the enzyme, it switches the enzyme to its off conformation, its off shape. And by doing that, it inhibits the active site. So this would be an example of allosteric regulation where it's inhibitory. Okay. You also saw an example through non-competitive inhibitors. So a non-competitive inhibitor inhibiting an enzyme is allosteric regulation. Okay, so a non-competitive inhibitor would be an example of allosteric regulation. Just to give you two other examples, these are going to both be positive regulation. So what you have here is a multi-subunit enzyme, and specifically they call two of the subunits regulatory subunits because they're involved in regulating the part of the enzyme that actually has the active site. And the part that has the active site you call the catalytic subunits. And so what they're showing you here is that when the regulatory subunits are bound to the catalytic subunits, they're inhibiting the enzyme from catalyzing the reaction. But then if you have the small molecule, cyclic AMP, if it binds to the regulatory subunits, it causes them to release the catalytic subunits, and now the active sites on the catalytic subunits are available and can catalyze the reaction. Okay, so that's an example of positive allosteric regulation. Another example of positive allosteric regulation is down here. And what you have is, again, a multi-subunit enzyme, but each of these subunits is identical, and each of these subunits has an active site. And again, there's two conformations for each subunit. This is the off conformation, and then this is the on conformation. And what happens in this form of allosteric regulation is when substrate binds to one of the subunits and the one subunit flips to its active form, that causes the other subunits to also flip to their active form. So the allosteric part is the fact that the binding here affects these three other active sites so that they're now active. Okay, do you guys have any questions about allosteric regulation? Yep. So, I mean, it can happen, well, it can happen in the cytosol, but it can happen anywhere you have enzymes. But so not all enzymes are regulated allosterically. So some are regulated through the other forms that we're going to talk about. And actually some, there are examples of enzymes that are regulated by multiple ways. So there's some enzymes that are both allosterically regulated and also regulated through the addition and removal of phosphate groups. Okay, so the next way that we're going to talk about in terms of regulating, enzyme activity is through the addition and removal of phosphate groups. And this is the form of regulation that's going to involve kinases and phosphatases. So one thing that can happen in this form of regulation is you have an enzyme and it's off, and then the addition of a phosphate group to the enzyme activates the enzyme so that now it's on. And then the kinase's involvement in this step is that the kinase would be the enzyme that adds the phosphate group to this enzyme and activates it. So you have a kinase come along, add a phosphate group that activates the enzyme, but then the same enzyme that's now on could be deactivated and returned to its off state by a phosphatase. So remember we said a phosphatase is an enzyme that removes phosphate groups, and so this phosphate group that was added by the kinase can be removed by a phosphatase, and that can switch switch the enzyme on and off. The nice thing about this form of regulation, and this is also true of allosteric regulation, is that it's reversible. So you can have an enzyme sitting in the cell, and without actually destroying the enzyme so that you would have to make more, you can turn the enzyme on and off when you need it on and off. So it's a reversible form of regulation. So is allosteric regulation. Okay, and there's other enzymes that act in the opposite way. So you're going to have other enzymes. I'll just call this one E2. And when it has the phosphate group added to it, it is off. And then when the phosphate group is removed, that is what turns it on. So in this situation, the phosphatase would be responsible for turning the enzyme on, and the kinase would be involved in turning the enzyme off. Okay. Do you guys have any questions about how the addition and removal of phosphate groups can affect enzyme activity? Okay. So then the last way that we're going to talk about in terms of regulating enzyme activity is called proteolytic activation. In contrast to the first two forms that we talked about, proteolytic activation is not reversible. So proteolytic activation is not reversible. And it only involves turning the enzyme on. So proteolytic activation involves obviously activating the enzyme. And what proteolytic activation is, or what happens, is proteolytic activation is whenever the breaking of covalent bonds within the enzyme activates the enzyme. So if you, one of the enzyme names you didn't need to know was proteases, and a protease is a enzyme that catalyzes the breakdown of proteins. It does that by breaking covalent bonds. And so proteolytic activation is whenever the breaking of covalent bonds activates the enzyme. So as an example of this, you don't need to remember the specific example for this class, but the inactive enzyme here is called chymotrypsinogen. And then what happens is when these two amino acids get cleaved out, you're breaking covalent bonds, you also cleave out these two amino acids, and when you cleave out those amino acids, that converts the enzyme to its active form, which is called chymotrypsin. Okay, and this specific example is found in your bodies, and so chymotrypsin is an enzyme that's located in your small intestine, and it breaks down proteins. And one of the things that happens is you make chymotrypsinogen inside the cell, and then you secrete it to the outside. And the nice thing about that is you make chymotrypsinogen inside the cell, and it stays inactive because it's chymotrypsinogen, and it only gets activated once it's outside the cell, which is useful because this enzyme breaks down proteins, so you don't want it active in the cell, breaking down the cellular proteins that are needed. You don't want it active until it gets outside of the cell. And so once it gets outside of the cell, there's another enzyme that cleaves these amino acids and activates the enzyme, and now it can catalyze the destruction of proteins. Okay, do you guys have any questions about proteolytic activation?
So at the proteolytic activation, like, how are they further regulated or compartmentalized programs that?
What do you mean?
Since they're just switched to one, I mean, what if they start producing? You stop that.
Well, I mean, one way is to actually just destroy the enzyme. But the enzyme, even though it's proteolytic activated, may be regulated in other ways that turn it on and off.
Okay.
I don't know any specific examples for you. Like chymotrypsinogen, I think is only regulated through proteolytic activation. So.
Well, yeah, that helps.
Okay, do you guys have other questions about proteolytic activation? Okay, so the last thing that we're going to talk about, is it the last thing? Yes. The last thing that we're going to talk about is how you can organize enzymes into what are called metabolic pathways. So what we're going to do is this, it says biochemical pathway at the top, but a biochemical pathway is the same thing as a metabolic pathway. So this is going to describe to you in general what happens during a metabolic pathway, and then we'll talk about it and talk about two types on the other side.
Organisms contain many different kinds of enzymes that catalyze a variety of different reactions. Many of these reactions, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, a substrate is converted into a product by the first enzyme in the pathway, and the product of the first reaction then becomes the substrate for the next reaction. The product of the first reaction then becomes the substrate for the second enzyme. The sequence of reactions continues until the final product is made. When a biochemical pathway is functioning, the initial substrate is continually converted to the final product through the series of steps in the pathway.
Okay, so essentially a metabolic pathway is simply a set of reactions. So it's a set of reactions where each reaction within the set is catalyzed by a different enzyme. Okay, so it's a sequential set of reactions where each reaction within the set is catalyzed by a different enzyme. That's all a metabolic pathway is. So there's a lot of cases where you don't go directly from the reactant that you're given to the product that you want to make. That can't happen in one step. And so you need to take the initial molecule that you make and tweak it in small ways along multiple reactions into a final product. And then it's tweaked in different ways by each enzyme that acts on it. Okay, so you don't go from reactant to the product that you want in one step. It takes a sequential set of reactions, and then each reaction within that set is catalyzed by a different enzyme. Example of metabolic pathways that we'll study in this class are going to be glycolysis. So in glycolysis, you take glucose. You don't need to memorize this detail for right now, but in glycolysis, you're going to take glucose and you're going to break it down into what are called pyruvate molecules. And that pathway doesn't happen in one step. It's actually 10 sequential steps that you go from glucose to 2 pyruvate molecules, and each step is catalyzed by different enzymes. So you have 10 enzymes that each act in a specific order to produce 2 pyruvate molecules from 1 glucose molecule. And so this is just showing you another example of what in general happens in a metabolic pathway. So enzyme 1 takes the reactant, the initial substrate, converts it into product B, and then product B is the substrate for enzyme 2. So B binds to enzyme 2, enzyme 2 converts it to substrate C, and substrate C ends up being the substrate for enzyme 3, and then enzyme 3 acts on substrate C to produce the final product. Okay, and that's essentially what happens in all metabolic pathways. You're going to tweak the molecule in little ways or big ways as it moves along the pathway. So there's 2 kinds of metabolic pathways. There's catabolic pathways, and then there's anabolic pathways, and the gist is on the board there. So in a catabolic pathway, you're taking a complex molecule and you're breaking it down into simpler components. So anytime you take something complex, break it down into simpler components, that's a catabolic pathway. Glycolysis, which I just described to you, is a catabolic pathway. So you take glucose and you break it down into 2 simple pyruvate molecules. And so another thing you should know about catabolic pathways is that they often release energy. So a catabolic pathway often releases energy. So the example that I have for a catabolic pathway up there is cellular respiration, and glycolysis is just one part of cellular respiration, but cellular respiration breaks down organic molecules. That releases energy. This particular pathway uses that energy to make ATP. Okay, so catabolic pathways, breaking stuff down, and these types of pathways often release energy. On the flip side of that, you have anabolic pathways where you're taking simpler components, combining them to make a more complex molecule. So in anabolic pathways, you're building something or making stuff. And as an example of that, I have protein synthesis on there. And also in contrast to catabolic pathways, anabolic pathways are usually going to require the input of energy. Okay, so anabolic pathways are usually going to require the input of energy, and that's true for protein synthesis. So protein synthesis requires the addition of energy in order to synthesize a new protein for the cell. Okay, do you guys have any questions about metabolic pathways or the two types of metabolic pathways that we've talked about? Okay, so the last thing, this is related to metabolic pathways that we're going to talk about, is feedback inhibition. And feedback inhibition is a way of regulating a metabolic pathway. So feedback inhibition is a way of regulating a metabolic pathway. And this animation will describe how feedback inhibition can work, and then I'll also describe it when the video is done.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
Okay, so essentially what you have occurring in feedback inhibition is one of the products of the pathway goes back, that's the feedback part, and inhibits an enzyme that acts earlier in the pathway. Okay, so in feedback inhibition, you have a product of the pathway going back or feeding back and inhibiting an enzyme that acts earlier in the pathway. So in the specific example that they gave you in this animation, the final product inhibits the first enzyme. So the final product goes back, binds to the first enzyme, and inhibits it, and in that way that's feedback inhibition. Okay, some nuances that I don't care if you know or not, just to clarify a few things, is it's not always the final product that feeds back. It could be an intermediate product that feeds back and inhibits one of the earlier enzymes, and then it's not always through non-competitive inhibition, because this would be an example of a non-competitive inhibitor. There's examples of feedback inhibition where it happens through competitive inhibition. Okay, so two nuances I don't really care if you know or not, but just so you realize it just doesn't happen this one way. One thing I do want you to know is why feedback inhibition is important. And one thing that it does is it prevents the overaccumulation of product. So it's wasteful in terms of energy for the cell to produce more than it needs. So the feedback inhibition is a way of inhibiting or shutting down the pathway once you've made enough of the product that you need. That happens so once the product reaches a certain concentration, you inhibit all of enzyme 1 in the cell, and that's going to shut down the whole pathway. So feedback inhibition shuts down the whole pathway, and again, that's going to be useful for preventing the overaccumulation or producing too much of the product. Okay. Do you guys have any questions about feedback inhibition? With feedback inhibition, I'll also give you two examples when we study cellular respiration. And just one example is that one of the products of glycolysis is ATP. So I told you glucose is broken down into pyruvate. That releases energy, and during that pathway, it's used to synthesize ATP. ATP turns out to be a non-competitive inhibitor of one of the enzymes that acts earlier in the pathway. So that would be a way of shutting that pathway down once you have enough ATP for the cell. Okay, if you guys don't have any questions, we'll take our 10-minute break now. And then what we'll do is we'll start with, when we come back, with the lecture questions for this topic, and then we'll move on to cell communication.